So thank you so much, Michael, for being here. We don't want to steal any more time from you. We look forward to your talk.
Awesome. Thanks, Matteo, for the kind introduction. Give me just a second to share my
Sure.
screen.
No worries.
And can you see my screen now with the presentation on?
Yeah, it's perfect.
Awesome. Cool. Then let's move this aside. Okay, awesome. Yeah, thanks, Matteo, for the introduction. Thanks, Mutz and Jiwang for organizing this, and Jiwang also for the invitation, for yeah, also everyone else who joined now, I think from different parts of the world, different time zones. Yeah, thank you for taking the interest in our work, and I'm very happy to showcase what we've been working on over the past, yeah, like almost 10 years now in the Julia Forel lab at ETH Zurich, where we tried to engineer E. coli such that it can actually grow on methanol and also turn it into chemicals. And today I'd like to highlight a bit the motivations behind this project, and also what kind of progress we've made over the past, yeah, couple of years towards this goal. So yeah, without further ado, just a bit of motivation, but I think here I'll be preaching to the choir mostly. Um, the petrochemical industry, so far as it operates today, is a major driver of climate change. We're utilizing about 500 million tons of oil equivalents annually. So this is coal, natural gas, oil taken together to produce all sorts of amenities of daily life, such as polymers, solvents, paints, you name it. And while all those products are essential for modern life, at the same time, they emit about 3.1 gigatons of CO2 equivalents annually, which is equivalent to about 6% of global emissions. But this is not even accounting for Scope 3 emissions. So the global impact likely is still a lot bigger. Now, um, yeah. A problem with the chemical industry and why its emissions are so hard to abate in general is the fact that we utilize fossil feedstocks, so we emit a bunch of greenhouse gases during feedstock extraction, but also we embed the carbon that's in oil, natural gas, etc., into our chemicals. Meaning that at the end of their lifetime, when we dispose of them, this embedded carbon gets released. And this may be through incineration, directly towards CO2. It might also be through biological decomposition, which a lot of times will also release CO2 and also methane. And this being the outset, I think there is some agreement that in general fossil feedstocks will need to be phased out to really enable zero emission chemicals in the future. Now, one vision for phasing out fossil feedstocks starts by producing chemicals directly from greenhouse gases. And the way to do this is first turn greenhouse gases into what's then called green methanol, and then use this as the new currency really of energy and also carbon equivalents to then produce chemicals from green methanol. And because we've started with greenhouse gases to begin with, once we dispose of our green chemicals and turn them back into CO2 or methane, we should end up with a closed carbon loop and really not be contributing anymore towards climate change by utilizing and also producing green chemicals. Now, green methanol today can be produced through two major pathways. One is we start out from biomass. In this case, it's generally called biomethanol, or the alternative approach is starting from CO2 point sources or methane point sources, and also going forward, probably directly from CO2 from the atmosphere, and turn that into green methanol together with renewable energy and green hydrogen. Now, green methanol itself is still pretty young, but today we're producing globally about 700,000 tons of green methanol annually, but this number is set to ramp up dramatically over the next couple of years. So if you look at, for example, 2027, we expect a global production to be at about 20 million tons annually. And just to put this a little bit into perspective, the total gray methanol market today is about 100 million tons. So this being a rather young market and technology still, this will make a dent compared to global methanol production. Now, the question then is really like, how do we unlock green chemicals utilizing this green methanol? And we are a biology lab, and I'm a biotechnologist. So the lens that we view this problem through is one of fermentation. And green methanol has a bunch of advantages also for fermentation. One major one is that utilizing green methanol, its production, really is independent from agriculture. And because we're not dependent on agriculture, producing our substrate for bioproduction requires lower land use, lower water use, is overall much more efficient when it comes to nitrogen use, and with that are connected then less eutrophication, and also there's good data on that we could also have an overall much lower impact on biodiversity utilizing this as a feedstock compared to conventional biotechnology feedstocks. And then also from an economics perspective, it also does not suffer from seasonality the same way conventional biotech feedstocks in a lot of places around the world suffer. Because, for example, if you look at Central Europe, we only grow our crops in summer, and then for the rest of the year, we actually don't have that much biomass to continue working with in the winter. Also, from a purely fermentation perspective, methanol as a substrate can be quite advantageous just because it is more reduced on a per carbon basis than sugar. It enables higher theoretical yields for certain compounds, for example, aromatics. Additionally, there's just a lot less out there in nature that can grow on methanol in the first place, which then enables processes that require lower sterility overall and might make in the future the whole process more economical. Another point I'd like to highlight here is also something that contrasts bioconversion of methanol to a lot of chemical processes, and that's that you can utilize dilute and also impure methanol streams for bioproduction, just because we don't need 99.9% or 90+% methanol concentration to enable growth on methanol. Now, the question then, of course, is what kind of host organism do we want to use for methanol conversion to green chemicals? And then if you look into nature, there are certain organisms that can grow on methanol as a carbon and energy source. They're generally called natural methylotrophs, methylotrophs being any organism that can grow on methanol. But so far, there's compared to other organisms, only limited industrial experience. So the angle from which we came from for this project was to flip this coin around and ask, okay, but there are organisms already out there that we know how to run industrial processes with. And the one that we picked was E. coli. We can utilize it to produce all sorts of chemicals quite efficiently, but of course here the problem is that it can't utilize methanol in the first place. And this was then the first defining question of this project. Specifically, how do we teach this old dog, or really this old horse, a new trick? And this new trick being utilizing methanol for growth and then later on also for production. Now, to establish methanol assimilation in E. coli, we looked through a variety of different pathways that are out there in nature, and then eventually settled on the ribulose monophosphate cycle for several reasons. But the two major ones was that, first of all, the rump cycle, for short, is the most efficient known methanol assimilation pathway out there in nature. And then also, if you look at E. coli metabolism, which is highlighted here, you realize that in principle, you really only need to express 3 genes to enable the rump cycle, and with that, methanol fermentation, in principle at least. And those three genes encode one methanol dehydrogenase, which oxidizes methanol to formaldehyde, then a hexulose phosphate 6 synthase, which condenses formaldehyde with the acceptor molecule ribulose 5-phosphate to hexulose 6-phosphate, which is then isomerized towards fructose 6-phosphate. And fructose 6-phosphate is already a metabolite that is very common in E. coli metabolism just because it's central to glycolysis. Now, when you run this pathway, the stoichiometric product is pyruvate, but it is also essential that the acceptor molecule for formaldehyde is replenished at all times, because once we deplete ribulose 5-phosphate, the entire cycle would break down. Now, when we express those three genes in E. coli, we could actually observe methanol assimilation into central carbon metabolism. However, and this is probably less surprising, we did not observe any growth on methanol. And just to give you a little bit of an overview for the reasons why this might not have been possible from the get-go, I'd like to introduce you to some here. One is that methanol oxidation might just be too slow. We know that methanol dehydrogenase is not a great enzyme, so that might be a key reason. Then also the first intermediate of the pathway, formaldehyde, is highly toxic to all sorts of cells, and E. coli here is no exception. And then the last point I'd like to stress here, and I kind of touched upon this on the previous slide already, is that we have this cycle, meaning that if any of the intermediates are being depleted, we will break the cycle just because we cannot replenish the acceptor molecule ribulose 5-phosphate anymore. And then E. coli has never seen the rump cycle before, nor has it seen methanol before. So it's rather unlikely that all the branch points away from the rump cycle will be regulated such that it can actually enable growth on methanol to begin with. And because this list is so long and the dimensionality of those problems seemed so large, we deemed rational engineering.
Yeah.
Sorry?
Sorry about that. No, go
Okay,
ahead, Michael.
everyone hear me? And okay.
Yep.
Good. Yeah, so we deemed rational engineering infeasible to really solve this problem. So rather than approach this problem through a purely engineering approach, we decided to have a look at evolution and see if evolution can solve this problem for us. But of course, every student learns during their first semester that for evolution to work, you need to have growth in the first place, but we didn't have any growth on methanol. Also, you need to have the right kind of selective pressure to really push your population towards better growth on methanol. So those were the two factors we needed to work ourselves around. And we approached the whole growth issue with a concept that we dubbed methanol dependency. And really what we did with methanol dependency was to ask the question if we can modify E. coli such that it loses its ability to grow on a given co-substrate, here called X, but whose non-growth phenotype could be rescued by assimilating methanol and then forming not all of its biomass from methanol, but having to form part of its biomass from this compound, albeit a very small amount to begin with. Now, to systematically approach this problem, we modeled E. coli metabolism using flux balance analysis, which is a tool which really allows you to comprehensively model all the reactions that are known in E. coli metabolism. And with this tool, we screened about 11 co-substrates, about 240,000 genetic backgrounds, which then totaled about 2 million conditions using this approach. And then we had a list of potential candidates of combinations of genetic modifications plus co-substrates. And one I'd like to highlight here is a knockout of the triose phosphate isomerase gene, which is essential to glycolysis together with the co-substrate pyruvate. When we tested a strain of E. coli that had this deletion in its triose phosphate isomerase gene, we found, as predicted through our computational modeling study, that indeed it could not grow on pyruvate alone. However, when we then additionally supplemented the medium with with methanol, we could actually recover growth in this medium, which then solved the problem for us that we had a strain now and a system in which we had growth that was dependent on methanol assimilation into central carbon metabolism. Now, to optimize evolution towards increased methanol uptake over time, we settled on a chemostat system. The chemostat really is a bioreactor that continuously drips fresh medium into the bioreactor while pumping out all of the excess medium. And this system allows you to set up an environment that is at the same time pyruvate limited and also methanol replete. Meaning that in this kind of environment, we'd expect any mutant that has a benefit, that has improved capability of methanol assimilation to outcompete all of its siblings. And this was a system, and it looked very promising to begin with. It was still quite the hassle to then really operate the system for longer periods of time, just because contamination, etc., becomes an issue if you run a system, biologic system, for, say, more than a year. Nevertheless, this was the system that we then utilized for a long-term adaptive laboratory evolution experiment, and we ran this system for about 250 generations, which took about 15 months. But after that time, we were then able to isolate a population from the chemostat that grew on methanol alone in the medium, albeit with a rather slow doubling time of around 60 hours. We then transferred this population into shake flasks and undertook another serial dilution experiment on top of it, and then were able to isolate individual clones from this population that grew with a doubling time of 4.3 hours after about 1,200 more generations of evolution. And this is kind of the point that we published this year. However, since we've continued our experiments, and now our strain actually grows with a doubling time of about 3 hours. And we don't think we're quite at the limit yet of this strain that we've developed. And maybe to also put this a little bit into perspective, we have this really artificial E. coli strain now, growing at a doubling time of about 3 hours, which is faster than a natural methylotrophic yeast, such as Comagidea paphi, and also a bit faster than, for example, Bacillus methanolicus, if you grow it at the same temperature. Bacillus methanolicus is still faster. For example, if you grow it at 50 degrees Celsius, we can't do this with E. coli because then, of course, E. coli will die, unfortunately. Nevertheless, conclusion for us, okay, now we actually have a system that is still an E. coli strain, can utilize methanol, appears quite efficiently. And this is now the foundation for all of our future work to build on. And building on top of this strain, we next set out to really show that we can still produce utilizing this E. coli strain. And there's really two big promises that our E. coli strain made when we set out with this product. And one was, okay, we're utilizing E. coli, so we want to reutilize everything that's known about E. coli for this strain. So production pathways, production plasmids, all the genetic tools that are available. And also we wanted to see if our E. coli strain still behaves nicely in a bioreactor setting. So if like basic scale-up is still possible with our methylotrophic E. coli strain. Now to test this, we first looked at 4 different branch points in E. coli metabolism and decided we wanted to show production from all of those branch points. The first being pyruvate, the second one acetyl-CoA, then the TCA cycle, and then lastly, we also wanted to test production from what is called the coarse-mate pathway, and the coarse-mate pathway has phosphanol pyruvate as a precursor, as well as erythrose 4-phosphate. And those pathways generally allow you to produce a variety of different compounds that are involved in cosmetics, packaging, resins and dyes, pharmaceuticals, and the list goes on. And we picked 4 specific ones. One is lactic acid from pyruvate, which has a huge market for food service ware and packaging. Polyhydroxybutyrate is also a bioplastic. then itaconic acid is just a base molecule for lots of different compounds in the chemical industry. And then p-aminobenzoic acid, for example, is used as a structural ingredient for lots of pharmaceutical formulations. Now, we first tested production of lactic acid from pyruvate. The reasons being that pyruvate is the stoichiometric product of the Rump cycle, so we'd assume good production of the precursor molecule. And the production pathway is rather short. It really just requires the overexpression of one gene. And when we tested this, we observed production of about 300 milligrams per liter of lactic acid, which was nice. We then tested polyhydroxybutyrate production, which is a bit more complex in E. coli. It worked. Titers in this respect could still be improved, but nevertheless, it showed it was possible. Then itaconic acid is being produced from the TCA cycle at good titers too. And then lastly, we produced p-aminobenzoic acid from the coarse-mate pathway, which is an aromatic, which is also a compound that has higher theoretical yield from methanol than it does from glucose. And that worked really well too, wrapping up this production package for us. And what our learnings from this one, if we compare this then to production from glucose utilizing also E. coli or also other methylotrophic organisms for the same compounds, which was really interesting for us to see, is the fact that even though we have this highly artificial system now, and we didn't improve any of the production strains that we had, we really just plugged in the genes that are necessary to complete the pathways. The titers that we gained and also the yields that we gained are very comparable also to an E. coli strain that grows on glucose, and also a lot of times better than if you use a natural methylotroph. Yeah, having this strain in hand, we then next asked if we can do small scale up of our platform in a bioreactor. To this end, we ran a fed batch 1 liter system and first tested if growth to high cell density was still possible. High cell density is important for improved production because the more cells you have, the higher production you expect at the end of the day. And when we tested this, we achieved optical density of about 100. It's equivalent to about 22 grams cell dry weight per liter, which was really nice. What was also nice was that we're pretty confident that we could even increase this cell density. It was mostly a limitation at this point of our hardware that we have available, specifically that we could not increase oxygen transfer further. And yeah, having this in hand, what we next tested was if this translated to improved production, in this case of itaconic acid. And gladly, we could observe that the increase in biomass resulted in a proportional increase of itaconic acid production in this case. In this case, about 10-fold, achieving titers now in like the low grams per liter scale for itaconic acid, which was really promising. And yeah, this is where we are at the moment with our strain. I'd like to give you a brief overview where we'd like to go to next. So one thing I mentioned was that our E. coli strain is a result of an adaptive evolution experiment, and it also acquired a hypermutator phenotype throughout its evolutionary phase, meaning that it fixed about 1,200 mutations now. But we're quite confident that really we only need a handful, maybe 2 handfuls worth of mutations. But to figure out which mutations are actually important, we are now running evolutionary replay experiments where we expect to produce independent lineages of methylotrophic E. coli strains. We can then compare the mutations that fix in all of those E. coli strains, and then compare those and see which ones fix repeatedly, and from there on out we should be able of engineering an E. coli that is actually genetically defined, hopefully with a lot less mutations than the current iteration has. And then also would like to improve production going forward. The aims are to achieve greater 10 grams per liter at about 80% yield. This is ongoing, and with that, I'd like to already come to the end of my presentation. I'd really like to acknowledge Julia Forhold, out of whose lab this entire technology stems, and she had the grit to stick through all the ups and downs of this project. It's been going on for almost 10 years at this point in her lab. And I'd like to highlight Philip Keller, who'd been instrumental in really coming up with the evolution experiment, and Timothy Pradley, who co-authored the production paper with me, and Lars Büchel, who is now running all the chemostats for us. And yeah, one last point I'd like to mention here. So we are planning to explore commercialization with the technology that we have right now. So if you have deeper insight into the chemical industry, I'd like to chat. If you want a partner, I'd be curious. If you're an investor, it would be cool to exchange, or if you have specific knowledge about like IP, carbon pricing, or you just want to challenge our assumptions, I'd be yeah, very glad if you'd reached out. And yeah, with that, I'd be happy to answer any questions you might have.